Hi everyone, and welcome to our exciting screencast all about genetic mutations. So let's start talking about how mutations occur. Now, mutations are caused by mutagens, and mutagens can be a type of energy, like an X-ray, or a type of chemical. And of course there's many others which we'll talk all about in the cancer unit. So mutations can occur naturally just by a common mistake that's made in DNA replication, or the environment can cause it. Some other things are foods, plastics, pesticides. And there's two types of mutations, gene mutations and chromosome mutations. We'll start by talking about gene mutations. So a gene mutation is also called a point mutation, and that's when the only thing that's changing is one or sometimes a few more nucleotides, letters, in a single gene. There's three types, so you can either have additions, deletions, or in this case, a substitution. So let's talk a little bit about what we already know. We already know that DNA, when it replicates itself, you need complementary base pairing. Those complementary bases join the original strand to make a whole new strand, and mistakes can be made during this process. You can also have mistakes when you're copying DNA to mRNA before a protein is made. So here's your mRNA, and mRNA is translated into an amino acid chain, and that creates the proteins. When you're copying DNA, one of three things can happen when you're talking about gene mutations. You can get a substitution mutation, where again just one letter is substituted, the wrong letter, and it's usually not a very bad mutation. You can also get a deletion, where one letter is left out, or an addition where an extra letter is put in. Both of those are quite serious mutations. So let's first discuss substitution. If you look here, GUU is the mRNA code that's supposed to be copied, and it codes for the amino acid valine. If you took that G and turned it to an A, you'd get a completely different amino acid. You'd get isoleucine, but only one amino acid is changed or affected. So let's look at deletion. If that G were deleted, then the amino acid code, instead of being valine, would be phenylalanine. And if you look, all of the protein is going to be altered after that nucleotide mutation, because after the mutation, every single amino acid that's coded for will be wrong. And if you look at an insertion mutation or an addition mutation, CGC is correct, so you still have arginine, but an extra A is added before the G, and so instead of having GUU, you have AGU. Instead of UCC, you have UUC, and again, all of the amino acids will be different after that mutation. Start by talking about substitution. Severity of the mutation, or how bad it is going to depend on which amino acid is changed. So again, it's just one letter that's changed. If you think of it like a sentence, instead of saying dog, you're saying dot. One base is changed. The sentence still makes sense a little bit because only that one letter is changed, and in the case of proteins, only one amino acid is wrong. If that amino acid doesn't have any significant R group, the change won't be significant. If you think about the three letters on an mRNA coding for one amino acid, you look at UAU, which is the correct one, it makes tyrosine. If you change the G, you make aspartic acid. If you change the A, change the C, oh, good news, you still get tyrosine, that's great. But if you change that last letter to an A, you stop the protein from being made. Same with if the last letter is a G, not good news. So the severity depends on where the change is. Let's use this as an example. If your original sequence is like this, and that A is changed to a G, you still have GLN for your amino acid, so that's good news. Even though there was a mutation, you get the same amino acid. And if you change UCU to CCU, you get one different amino acid. So that's not too bad, and depending again on that R group. If you change this amino acid to an UAU to a UAA, no protein will be made after that mutation. An example of the substitution is sickle cell anemia. So normally with sickle cell you have GAG, and with this mutation you have GTG. So one tiny little error gives you, instead of glutamic acid, you get valine. Now that turns out to be really bad news, because that R group in glutamic acid was really important, and now your hemoglobin isn't going to fold properly. So what you get is sickle cell anemia. With sickle cell anemia, instead of having perfectly rounded donut-shaped red blood cells, you get these really pointy, stiff blood cells, and they cause traffic jams in the arteries and veins, and that is not good news for your body. And that's because your body doesn't get enough oxygen where it needs to, and that can cause some serious problems. Now, in Africa and in other regions in the same latitude of the world, lots of people have sickle cell anemia. The question is why? The reason is because malaria doesn't like blood cells that have been sickled. And so more people are successfully surviving the malaria attacks if they have sickle cell. Now that's only true for the carriers of sickle cell anemia if they only have one bad gene, because some of their blood cells will be sickled and some won't, and malaria won't attack them. The other two types of genetic mutations are deletions and additions. Now deletions, again, when you get one letter left out, one nucleotide left out, and it's very serious because all the amino acids after the deletion will be wrong, so the shape of the protein and the function of the protein will be incorrect. So if you think about it as a sentence, complete sentence will be changed after that deletion. So if you leave out the boy, the sentence doesn't make sense anymore. And of course, when you're talking about proteins, the protein will just be wrong. It'll be the wrong shape. It won't twist properly. It won't be able to do its job. Addition deletion is just as serious because you've got one extra nucleotide added. So again, the sentence will be completely wrong. Let's talk about a few genetic mutations then. This is a girl from India who was born with 8 limbs, 4 arms, 4 legs, and they actually did surgery to help her with that. Many animals have this problem: 2 heads, too many limbs, like this duck. Of course, here's a little baby with 3 arms. Kitties with more ears than they're supposed to have. And albinism is another genetic mutation, and it affects the protein melanin. And here's just a few examples. Dolphin is actually pink. So many animals have albinism. And in nature, it's not usually very good to have albinism because you stand out in the crowd. It makes you more susceptible to being attacked by predators, of course, depending on what your environment is like. Polydactyly is another genetic mutation. Poly means many, and these are people that are born with too many fingers or too many toes. Here's the fingers, and here's some patients born with too many toes. If you count this guy, he actually has 8 toes on both of his feet. Gigantism is another genetic mutation. You've probably heard of Andre the Giant, who passed away. These people have problems usually with their pituitary glands, and they have too much growth hormone when they're children and can be over 9 feet tall. You can see here is a picture showing you dwarfism and gigantism versus a normal person. And this isn't a real picture, but it gives you an idea. So you can see the mother and the son here. The mother is quite a normal size, but it's hard to get that perspective when the son is so large. Another genetic mutation or gene mutation is with the wolf man or wolf woman, and it's called hypertrichosis universalis, and it's a genetic mutation that basically makes the hair growth not work properly in your cells. So normally your cells would prevent hair from growing in certain places. It's called the werewolf syndrome, and 1 in a billion people get it, so it's quite rare. These are 3 sisters here. You can see a few people affected with the werewolf syndrome. So a few other gene mutations are cystic fibrosis. This causes problems with your lungs. Sickle cell anemia, we already talked about, causes problems in the blood, and Huntington's disease causes problems in the brain. At a certain age, around 40, you start not being able to think properly, similar to Alzheimer's. And not Alzheimer's is caused by a genetic mutation, but in this family, 10 out of 14 people were. And there's the Schwarzenegger gene. This is a myostatin gene. This is a bull that was born normally. It had way too many muscles because of a genetic mutation. Cancer is also caused by genetic mutations, usually by more than one genetic mutation. So the more you're around mutagens, the increased chance you have of getting cancer. Here's skin cancer, and when we talk about cancer, we'll be seeing a lot more. Okay, so let's talk about chromosome mutations. Chromosome mutations are quite different from genetic mutations because with a chromosome mutation, all or a part of an entire chromosome you'll be mutated or changed, and this affects more than one gene. It affects many genes, and so you have a bigger problem with a chromosome mutation, or benefit depending on how you think of it. Some chromosome mutations led to many evolutionary advancements. An example of a chromosome mutation is called crossing over. This is where a whole chunk of a chromosome will switch places with another. So you see here the crossing over of these two chromosomes, and that will cause pieces to be in the wrong spot. An example of this is with Cri-du-Chat syndrome. This is Charlotte, and she's got Cri-du-Chat syndrome. And what happens is a piece of the 5th chromosome is completely missing. So here's the 5th chromosome, and you see a chunk of it is absent. The reason it's called the Cri-du-Chat syndrome is when babies are born, they actually sound like their cat's crying. It's a very strange sound, and they end up with mental difficulties when they get older. Another type of chromosome mutation is non-disjunction, and that's a mistake that's made during meiosis. So with a normal meiosis, you'd get 2 copies of each chromosome in each cell. With non-disjunction, what happens is you can end up with 3 copies of each chromosome in some of your eggs or sperm and 1 copy in the other. Now usually when you have this happen, you get a miscarriage, because when there are mistakes in DNA, you usually miscarry. But sometimes the body will allow a child to be born with non-disjunction. An example of that is Down syndrome. So with Down syndrome, it's also known as trisomy 21. And basically, if you have Down syndrome, you have 3 copies of the 21st chromosome instead of having only 2. There's a karyotype of a person with Down syndrome. They've got 3 copies of chromosome 21. So Down syndrome occurs in about 1 in 700 births in Canada. And people that have Down syndrome, because they have 3 copies of the 21st chromosome, have 47 chromosomes instead of 46. And this isn't very common to young mothers. Between 20 and 24 years of age, it's only a chance of 1 in 1,450. But when you get older, over the age of 40, your chances get much higher of having a child with Down syndrome. And so usually you get sent, in this case, to have an amniocentesis check. So it's a test they do. They take some amniotic fluid out of your womb and check it for the chromosome disorder. Another example of non-disjunction is called Klinefelter syndrome. Now, Klinefelter syndrome happens when sperm or your egg carries an extra X chromosome. Usually the egg has 1 X and the sperm has either a Y or an X. But if this happens, you end up with XXY, and that's called Klinefelter syndrome. And you would be born a man, but you'd have breasts and you can't make sperm. And so here's a karyotype. You see the 2 X's and a Y. XXY gives you Klinefelter syndrome. When you have this disorder, you're quite tall, but you have a feminine physique. Your IQ is impaired. Your testes are quite small. They're atrophied. You're more prone to get osteoporosis like females are. You have breasts. You don't get bald as much, and you can't grow a beard. Turner syndrome is kind of like the opposite problem. So instead of having an extra sex chromosome, you're missing a sex chromosome. So instead of being XX or XY, you're just plain X. So you're missing 1 X chromosome. People born this way are female, but they're quite short. They have widely spaced nipples. One of their fingers is shortened. Their fingernails are quite small. They have brown spots sometimes on their bodies. That's called nevi. They don't bleed. They don't menstruate once a month. So they can't have babies. They're sterile. Their ovaries are quite small. They're underdeveloped. Their elbows are deformed. Their breasts don't develop properly. Their aorta are squeezed, and they have this fold of skin on their neck and very characteristic facial features. So they only have 1 X chromosome, and that causes a whole range of problems. Here's a baby born with that fold on her neck, and when they get a little older, it looks something like this. So very common when you have Turner syndrome to have this feature. So just to review, there's 2 types of mutations: gene mutations and chromosome mutations. So I know you're going to have some hot questions about this one. Make sure you come to class with all of your questions.